Yeah, these new technologies, the gene edition, also eles provide a way to not only to cut the DNA and allow the DNA do the repair or change one base or another, but also allow to insert some DNA fragments. This is the way that they produce the gene driver. Say they introduce a sequence. Once this sequence is in the genome of one, like in the diploid organism, if you have a diploid organism, just one sequence will convert another sequence in the other, or in the same locus, will change the sequence of the homologous chromosomes, the locus on the homologous chromosomes. So by doing that, you can impose to the population one trait that will be, as you said, will be exponentially spreading over the population very quick. So they believe that doing that, they will control, again, the word is control, a pest, like if you insert a gene for a male organism or a male insect, all of them will be male soon, and then they will not reproduce and you get rid of this. In fact, we have one experience that is not with CRISPR technology, but with recombinant DNA technology that was the mosquito, the Oxitec mosquito, that was quite similar in the fact that they try to kill one of the sex, but the percentage of the individuals that was killed was not 100%. Why not? Is because of the genetic background. The genetic background, I say the other genes, because if you have some insects that cross each other, they have a different genetic background. So there is some pleiotropic effects among genes. So maybe not all of them will be male. So if not only they will behave as a male, I should say, you have a room for crossing, and this will be also not only spread to the wild population, but also will not kill all the insects that cause one. This is one of the issues. The second issue that this cancer is under the evolutionary pressure. Thus, this sequence can be mutated, can be affected by epigenetic effects like methylation or something. In other words, they are not free of the evolutionary pressure on the sequence. And some of them also can cross with other individuals. And usually there are many bacteria that are attached to these insects. And some of them also exchange some genetic material. So, in other words, we are not sure where this genetic modification or the fate of this new, so where they will be, where they are going, who will be the limits of this modification. So we cannot control what will happen. And also, as far as we have experience, not with CRISPR gene drive itself, but with similar technologies, they are not 100% efficiency. efficient. So, in fact, I think the gene driver is a way to spread some genetic modification and without any control. This, in fact, we have no way to control. This is worse than to release one variety, because one variety produces seeds, you can take the seeds. But insects, they use insects. There is no way to recall an insect or do something else. So this is worse than release one variety. And this is, since we don't know what will happen in future, in different ways that this modification can be spread, so how we can forecast or take some measure to avoid some possible adverse effect in nature.